Right now, your body may be carrying a genetic sequence shared by fewer than 1 in 10,000 people on Earth. These mutations arrive hard-coded before birth, rare departures from the standard human blueprint. To find evidence of one, look at the upper part of your ear, right where the cartilage meets the side of your head. A pinprick-sized hole in this spot is known as a pre-auricular sinus. This timeline maps the development of facial structures from early aquatic vertebrates to modern humans. Evolutionary biologists trace this specific pit to a variation in the FOXY3 gene, identifying it as a remnant of a gill slit from ancestors that moved from water to land. While often overlooked, these small structural quirks are actually ancient biological data points still present in the modern genome. Another data point exists on your feet. If your second toe extends past your big toe, you carry a dominant gene for a structure called Morton's toe. Ancient Greek sculptors viewed this anatomical variation as a physical ideal. It is a detail preserved in the copper feet of the Statue of Liberty. You can also see this trait in the marble of Michelangelo's David, where it was used to signify classical perfection. This diagram illustrates how a foot with Morton's toe distributes force during a stride. This single dominant gene alters weight distribution across the arch and changes how the spine aligns during movement, often resulting in a naturally different gait compared to the standard foot. While some traits influence mechanics, others are vanishing entirely. By touching your thumb to your pinky and flexing your wrist, you can check for the palmaris longus, a tendon running up the center of the forearm. This muscle was once used for navigating forest canopies. Because humans no longer rely on tree climbing for survival, the gene responsible for this tendon is slowly deactivating across the species. With roughly 14% of the population now born without it, our bodies are actively phasing out obsolete mechanics while maintaining divergent physical systems. This shift is also visible in our skeletal structure. Individuals born without wisdom teeth are part of a rapid change in human bone architecture occurring within living generations. This wireframe tracks the transition from an early hominid skull to a modern one. A mutation in the MYH16 gene is responsible for both the disappearance of the rear molars and the physical retraction of the human jawline. 35% of the population is now born without these teeth. The speed of this transition proves that the human frame is still being edited in the present day. These edits extend to our sensory perception. Hereditary heterochromia, mismatched eye colors, is more than a visual curiosity. It occurs when the OCA2 and HERC2 genes distribute melanin unevenly. This results in two different irises within the same person. Because pigment levels affect how light is absorbed, these two eyes process low-light environments differently. The brain must constantly adapt to these two distinct streams of visual data. This chart compares standard vision with tetrachromacy. Most people have 3 cone cells, but some possess a fourth, represented by the right peak. As standard peaks fade, this fourth zone remains active. Tetrachromats see 100 million colors, a range screens physically cannot capture. Up to 12% of women may carry the gene for this fourth cone. However, since no standard optical test exists for this trait, most individuals remain unaware that they are seeing a version of the world others cannot access. They inhabit the same physical spaces as the rest of the species, while experiencing a significantly wider version of reality. In 2006, researchers studied a street performer in Pakistan who walked on hot coals without any physical reaction. He felt absolutely no pain. His condition was traced to a rare loss-of-function mutation in the SCN9A gene. The SCN9A gene regulates sodium channels acting as pain gates. In this mutation, those gates permanently close, preventing the signal from reaching the brain. When it misfires the opposite direction, the gate locks permanently open, flooding the brain with excruciating pain. By mapping these extremes, researchers isolated the mechanism governing how the human body processes physical suffering. Major pharmaceutical companies are currently using this genetic data to develop a non-addictive class of painkillers. These treatments are designed to target the sodium channel directly, blocking pain without affecting the rest of the nervous system. This targeted treatment was developed by studying the DNA of individuals born with these rare neurological profiles. Your body is a 3 billion-letter library of genetic code that is constantly being edited. The anomalies found in your own DNA are not random errors. They are the active prototypes for how the human species continues to adapt.